I want to start by thanking the organizing committee for putting together such a wonderful conference. Congratulations, James, and thanks for including me. Okay, so my talk will build on Rowan's talk earlier, and also it touches on some of the issues that were brought up in today's plenary lecture this morning, actually, by Professor Sir Peter Knight. Okay, so my group is interested in organic semiconductors. So we view them as quantum materials that are useful for harnessing energy. So there's an example of a solar cell there, right, made of organic materials. And also earlier in the conference, we also saw several different talks about the application of organic materials in OLEDs. So here are some examples of organic materials, right? Chemical structure. So the common theme here is that you have alternating single and double bonds, and that is very important for exciton delocalization and and coherences, as well as super transfer, a concept that was introduced on the first day of the conference. Okay, so we're also interested in organic materials because they have the ability to generate energy. So now I have a research program on studying how we can actually harness energy using organic materials and turn them into fuels, right? Particularly hydrogen gas. And I know Rowan gave a very good talk earlier about how we can actually improve solar harvesting using interesting processes such as singlet fission. So I also have a research program on that. But today I will tell you about our work on studying coherences in organic semiconductors. And Rowan gave a very good talk on the use of transient absorption spectroscopy. So the reason that transient absorption spectroscopy is quite widely used today, and it probably can be traced back to one of the seminal papers in the field published back in 2009 by I'm not very good at using this, by Greg Scholes, right, a native of Melbourne. So he demonstrated that we can use optically prepared coherences to give us a lot of insight into what actually happens in organic semiconductors that give these materials really interesting properties in different applications. So transient absorption, right, is required because we need good time resolution. So you need around about 100 femtoseconds or less. Most of the time you actually need for example, 10, 20 femtoseconds in order to resolve a lot of interesting coherent processes in these materials. So these coherences, right, they decay within picoseconds. So you need really high time resolution to actually resolve the oscillations, and I'll show these oscillations later on. So transient absorption is great because ticks a lot of boxes here. So the type of data, right, we typically get in the lab looks like this. So on the y-axis you have time, and then on the x-axis you have wavelength. So basically you have then the spectrum, and Rowan gave you quite a lot of examples of transient absorption spectrum, such as the one on the right. And if you then draw a line vertically, oh dear, okay, got to remember this, draw a line vertically, then you get to see what happens in time, right? So you get the sort of spectrum on the sort of transient on the left. And this is all coming from pumping using a single wavelength, okay? And the information about pumping exciting using other wavelengths is not here. So there is actually additional information you can get by just changing the excitation wavelength. So to do that, basically, what we need to do, obviously, is to get a second dimension, right, an excitation frequency dimension, and to give us further information. So in this case, right? We go to something that's quite well known in physics and in chemistry. We go to Fourier transform spectroscopy. Okay, so all we need to do really is to look at some basic examples. So this is in the undergraduate textbook, right? So you look at something like FTIR. So you have your light source, and then the light source is split into two different beams. And by delaying one of the beams, this one here, you can then get an interferogram. And upon Fourier transform of the interferogram, one then can get a spectrum that looks like this. And it tells you a great deal about what happens to the molecule in terms of its spectrum. So converting from time domain to frequency domain is all you have to do. So in this case, let me give you another example. So it turns out NMR spectroscopy is another example. So you have free induction decay, right? Radio waves decaying, and by Fourier transforming the radio wave interferogram, one gets important information about chemical or nuclear interactions. So in fact, this is very well known to biophysicists, right, or physical biologists who have used NMR, right, in a sequence of pulses to do two-dimensional NMR. And in this case, right, so people in the field of biophysics, for example, have used a technique like this to solve protein structures. I shouldn't say that, maybe peptide structure. So protein if the protein becomes too big, then it's very difficult to assign individual interactions. But the point is, as Jeff explained just now, by looking at the diagonal peaks and the cross peaks, one can then assign interactions. And by assigning interactions, that leads to a lot of interesting information, such as in this case, I'm solving the peptide structure. Okay, so in our case, we do experiment not in the radio frequency, but in the optical frequency. So we need, in this case, a two-dimensional spectrometer that's in the visible range. And it looks kind of like this, kind of ugly looking, very complicated, right? But let me just try to break it down to you because we've done some technique development. So it's actually very fun to look at. You have a series of beams here. So instead of a sequence of radio wave pulses, you have now optical pulses, right? And they're very fun to look at. And the timing of the optical pulses is important. And to have fine control of the optical arrival time of the optical pulses is the key here. So 3 optical pulses, and then then it generates a photon echo signal, and that signal is then interfered with our 4th beam, right? Just like the case of FTIR, you have a reference beam, and in the end, you end up with signals that you can use to Fourier transform to get you two-dimensional spectra. Okay, please go forward. Help. Okay, the heart of it, right, is actually fine control of the optical pulses, and this is our development here. So we actually use a pair of rotating optical flats. So we actually made the spectrometer a lot easier to align and a lot cheaper to implement as well. So basically, just simply by rotating the optical flats, we can control the timing of the optical pulses. Okay, I'll just go forward. All right, let me just give you a couple of examples that we've done, examples of what we've done with this spectrometer. So this is a work in collaboration with a very outstanding individual here. And so this work was done when James was a Ramsey Fellow at the University of Adelaide. So, sorry. In this case, we were interested in asking the question, when we take a molecular assembly, oh dear, I should have checked the slides. I'm supposed to have quite a molecular structure here. I hope, let me just go forward. Yeah, okay. sorry, the chemical structure did not come out well. I don't know why. But it's a molecular assembly, and it's basically made of a series of these molecules here. So the purpose here is to try to study the coupling, the interaction of a number of these units that are chemically linked together. And the purpose is to see whether we can understand the interaction between the individual chromophores, and to see whether there is coherent effect in these in this molecular assembly. So these are typical data. So you can see that I'll just draw your attention to two things. So please focus on this spectrum here, and then also the time, right? This is time of the evolution of the system is indicated on the lower right. So basically you can see that this peak here is breathing, right? It's expanding, contracting, and that's basically, that's quantum beats, okay? Those are quantum beats. So by analyzing by that time, analyzing the time domain signal, we can then get the important frequency, right, in the coupling of the chromophores. So in this case, we did the experiment on the light harvesting complex, right, this assembly of many different chromophores, and we analyzed the time domain signal, and then we also did the experiment on individual chromophores, and we got the two Fourier transform spectra here, and they look very similar. And in this case, right, our conclusion was that we were hoping to see interesting coupling between the chromophoric units, but in the end, we could not do that. And that's kind of makes sense, because our assembly is not like your light harvesting complex in nature. So it's the individual chromophores are actually able to rotate and to move. So the amount of coherent effect is probably not going to be very clear there. I have another example, but I think due to interest of time, I'll just quickly run to this slide here to show that when we look at another example and another molecule, right, with very well-defined vibronic progression, we can also begin to tailor our light source to then try to answer some questions in terms of coupling there. So in this particular example, we look at the spectrum, and then there is pretty substantial change in the shape of the peak. And by analyzing the change in the slope of this peak, we're able to resolve all these oscillations. And then by Fourier transforming the oscillations, we then obtain information about the coherent vibrational modes in the excitation. So in this case, this is a well-known Raman mode for this particular molecule that's called TIPS pentacene. All right, and we're able to simulate the data using a model, and the end result is that we're able to determine the strength of the coupling between the molecules. All right, I hope I've given you a sense of what we can do with a coherent light source, and what we can do with multidimensional spectrometer. And we can analyze these quantum beats, and we can actually obtain quite a lot of data. And finally, I'd like to thank the people who did the work. So Patrick was a was a postdoc in my group, and this is Patrick before he was involved in my project, and this is after. So apparently my project was very good for facial hair growth. This is Greg Scholes, and a lot of other researchers and collaborators, including James, and thank you for your attention.
Thank you, Tak. Questions? No questions out there? Oh, there's one. Yep.
Thank you, Tak. I didn't quite get what you learned from the change in the slope of the peak. Could you go over that again?
Yeah, so the slope basically represents the amount of the coupling. So the peaks, they undergo slight change in the coupling frequencies. So basically, so when it's more aligned to the diagonal, right, it has a particular slope, and then when it actually when it's vibrating, then it has more coupling with another molecule of another mode, then it changes shape. So basically, by analyzing that, we understand then all sorts of
The
different
Wang
couplings.
Rees factor out of that.
Correct. Yeah, the Huang-Rees factor of the molecule. Yeah.
Thank you.
Maybe I can ask a quick question. So that TIPS pentacene is a classic singlet fission molecule. Is that you're going to go in that direction? Right now, it's a monomeric thing.
Yeah, so we did most of those experiments all in isolated molecules, and we also did some experiments on aggregates,
Okay.
and we did not see a lot of differences.
Okay.
Yeah, that's quite surprising. So that remains unpublished because it was not super interesting, whereas understanding vibronic coupling was actually more interesting in that case. So unfortunately, singlet fission does not seem to depend on
Such
on
coherences.
yeah,
Okay.
a lot of the coupling. So it's probably quite similar in coupling to just individual molecules. Yeah, quite interesting.
Good. All right. Well, thank you so much again. Great talk, Tak.